Hello everyone, I'm Madison Marshall, a master's student in the MacIver Lab at the University of Toronto Scarborough. Thank you for tuning into my presentation today. I would like to begin by acknowledging the land on which we're gathering today virtually and on which the University of Toronto operates. This is the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. For thousands of years, Indigenous peoples have inhabited this land and cared for it. Today, this meeting place is still the home to many Indigenous peoples from across Turtle Island, and I'm grateful to have the opportunity to work on this land. By doing so, I give my respect to its first inhabitants. Today, I wish to introduce to you my research that I do here, under the supervision of Dr. Scott MacIver, on landscape factors influencing cavity nesting bee and wasp diversity in the city of Toronto. Cities like Toronto impact biodiversity, but they continue to be important for the study of ecology, for the conservation of ecosystem services, and for connecting people with nature. Unfortunately, nature in cities is impeded by urban land use. From buildings to neighborhoods, the amount of natural habitat varies, as do the species found in this habitat. Urbanization leads to the homogenization of the landscape, which subsequently reduces biodiversity. Heterogeneity is very important to biodiversity, since a heterogeneous landscape has more resources distributed in a mosaic across an area. Also, a heterogeneous landscape composition supports species that rely on more than one type of habitat, which permits their survival in areas where they otherwise would not. Studies such as Gwinnett et al. In 2019 have found that different taxa are not equally impacted across an urban landscape. Considering this, cities are important spaces to study taxa that provide important ecosystem services, for example, bees and wasps. Wasps provide biocontrol for pest insects and other arthropods, and bees, as many people know, are important pollinators of flowering plants. Howe et al. In 2017 suggested in their review that cities are important refuges for bees. Whereas last year there was a review on pollination diversity and pollination services by Wenzel et al., which found contrasting effects linked to differences in the type of urbanized area and the intensity of urbanization. One group, known as cavity nesting bees and wasps, seem to be abundant in cities. Naturally, these bees and wasps nest in places like hollow plant stems and holes in dead wood, but they have been known to take advantage of nesting space in the built environment, such as holes in brick and mortar, nail holes in wood, etc. This adaptation is likely linked to why studies have found that these taxa are often in cities and may be less impacted by urbanization than other groups such as ground nesting bees and wasps. One urban land use which has proven quite valuable to biodiversity in cities is residential gardens. These spaces occur along gradients of urbanization and heterogeneity, and as green spaces, they act as corridors to larger habitat patches such as parks and naturalized areas in the urban landscape. Alone, they also serve as primary habitat space for a diversity of species, especially plants and insects, making them an excellent place to investigate some of these, like bees and wasps. Considering all of this, we have two questions that come up. How does urban land cover impact species diversity or abundance of bee and wasp communities? And are bees less impacted by urbanization than wasps? Investigating the cavity nesting community collected at sites across the city, we will be able to determine emerging patterns in bee and wasp communities and understand the impacts of urbanization on these taxa to help contribute to their conservation, habitat protection, and ecosystem services. To answer our questions, we decided to test several hypotheses of how community structure is affected by urban land cover. We hypothesized that abundance, richness, and diversity of bees and wasps are negatively correlated with urban land cover, since impervious surface represents a lack of natural habitat and natural resources. We believe that there will be a positive correlation with landscape heterogeneity, measured here by land cover diversity, with the Simpson Diversity Index, since higher diversity indicates more niches available and with more equal distribution, as well as positively correlated with land cover evenness, our other measure of heterogeneity, since this represents equally distributed resources with no dominant land cover. And finally, we also believe that there will be a positive correlation with green space, both open and closed. I'll explain those further later. And this is because these belong to natural and semi-natural land cover. To answer our research questions, we partnered with the David Suzuki Foundation to run the BIMBY, or Bees in My Backyard, project. This community science pollinator project engaged residents from across the Greater Toronto area to collect and observe cavity nesting bees and wasps using nest boxes hosted in their home garden. Cavity nesters readily nest in these man-made nest boxes, and as you can see in the figure below, the nest boxes hold cardboard tubes of 3 different diameters protected in a PVC pipe, and various species of these cavity nesters will choose to build their nests inside each of those individual tubes. The nesting behavior and central place foraging of these species make them an ideal study organism in urban areas, since these nest boxes only need to be set out once and they encapsulate the community over an entire season. And then tubes containing completed nests of bees and wasps can be opened for identification and linked to the unique area in which they were found. Nest boxes were also chosen because it is an excellent non-destructive sampling method, because we did release the specimens after identification, and this goes hand in hand with the greater purpose of the BIMBY project to foster awareness, curiosity, and urban conservation for these beneficial insects. As previously mentioned, sites were selected through the BIMBY project, using the home garden of each participant who registered for the year to host a nest box which sampled the cavity nesting community at their location. This resulted in 104 residential garden sites within the boundaries of the City of Toronto, as shown on this map, that got sampled from May to October in 2019. These could then be linked to spatial data analysis to uncover the links between cavity nesting community patterns and the land cover at each site. Once specimens were emerged and identified, they were then released, and counting brood cells, as shown here on the left, these nest tubes were opened and we were able to quantify abundance, richness, and diversity. We broke these down into groups, so we did abundance and richness for bees only and wasps only, as well as for total, which was bees and wasps together, as well as total community diversity, bees and wasps together. Diversity was calculated using the Shannon Diversity Index, and I'd like to mention that abundance, richness, and diversity were measured at the genus level, since all specimens weren't identified to species level due to destruction by parasites. Using open data sources from the City of Toronto, land cover data from 8 different cover classes were used to create 5 explanatory variables to test our hypotheses. There was open green space, closed green space, urbanization, and finally, land cover evenness and land cover diversity. Both evenness and diversity are measures of landscape compositional heterogeneity, with evenness being a bit simpler, the measure of the equality and proportion of each present land cover, and diversity being a bit more complex, measuring the number of different land cover types and their proportions. Each variable alone and in pairs were used for generalized linear models for bee and wasp abundance, richness, and overall cavity nesting community diversity. The land cover was analyzed at a 200-meter and 500-meter radius from each nest site, since these distances represent common spatial scales used in the literature, and they're linked to average foraging distances of cavity nesting bees and wasps. The best models were compared and selected using the Akaike information criterion. At the end of the season, we had collected a total of 1,406 nests containing 7,471 brood cells of 14 genera across our 104 sites. 6 bee genera and 8 wasp genera constructed nests, with bees producing more brood than wasps. The most common genera across our sites are Megachile and Osmia bees, as well as Tripoxylon wasps. The result of our modeling analysis of land cover within a 200-meter radius of the nest site showed unanimously that urbanization and land cover evenness are factors affecting cavity nesting bee and wasp abundance, richness, and diversity. As we hypothesized, urban land cover has a negative effect, whereas heterogeneity of the landscape, as measured here by land cover evenness, has a positive effect on cavity nesting community. Wasps are uniquely best modeled by a significant negative relationship with urban land cover. And moving to look at this at a larger spatial scale of 500 meters from the nest site, we found similar patterns within the community. The only difference being that the overall community diversity and richness was affected by urbanization and land cover diversity rather than land cover evenness. And also, the richness was best modeled by a positive relationship to land cover diversity rather than evenness as well. From our modeling analysis, we see that heterogeneity is supporting cavity nesting bees found in residential gardens, whereas wasps are being negatively impacted by urbanization. Wasps, as I've mentioned, are predators which hunt other arthropods, and they use these to provision their nest. We expect their prey to be found in forested areas and urban parks. When doing our landscape analysis, we saw that tree cover is significantly negatively correlated to urban land cover, which indicates that urbanized areas lacking trees are a threat to wasp biodiversity in city landscapes. As urban land cover increases, open green space is reduced as well, further reducing the potential hunting habitat of wasps. Surprisingly, though, urban green space was not a significant parameter in any of our models, and we didn't expect this because of the particular importance of urban parks and green spaces as habitat corridors which host flowering species for bee forage, as well as being potentially good spaces to host prey species for our wasps. A last thing I would like to point out here is that these models all had very low R-squared values, which tell us that bees and wasps cannot be explained by land cover alone in urban spaces. It's very likely that other factors such as microsite conditions like light and temperature, floral and prey resource distribution and abundance for bees and wasps respectively could also be analyzed and included as parameters in future models of this taxa. Coming back to our research questions, we see that urbanization has a negative effect on these taxa, with landscape heterogeneity being a positive factor influencing abundance, richness, and diversity. We also see that both bees and wasps are negatively impacted by urbanized land cover, but patterns in bees are best shown by a positive relationship to land cover heterogeneity, whereas wasp patterns are more closely linked to the negative effects of urbanization. The results of this study show that residential gardens are valuable for bees and wasps negatively impacted by urbanization. Our next steps are to characterize these spaces, being residential gardens, and investigate microsite conditions. This was our original intention for this project, but it did have to be pivoted due to COVID-19. We have identified here key parameters to include in other modeling studies of these taxa in cities. And considering the importance of land cover evenness, we have a few suggestions. We believe that heterogeneity should be incorporated into urban planning and development by including urban parks and forests around residential areas, as well as more effort into conserving existing trees and planting more trees to improve habitat for wasps in particular around residential areas. Since these landscapes are human dominated, it's essential that we continue to encourage community engagement related to bee and wasp conservation in the city, and we did this through the BIMBY project. Households we shared this research experience with have shared what they learned about bees and wasps in their backyards to a much larger network than we could have reached alone. On that note, I would like to extend my thanks to the David Suzuki Foundation for their contribution to the BIMBY project, as well as to all the community scientists who have been with us for over 2 years now. My biggest thanks goes to Dr. Scott McIver, who is an excellent mentor and advisor for this project and for the entirety of my master's research. A special thanks goes out to my lab mate Garland, who is essential to both the spatial analysis and modeling work for this research. Thank you all again for tuning into my OE3C presentation today. I hope you enjoy the rest of the research being shared by the presenters this year and have a great day.